Hi, I'm Jim Eubanks. I'm the Director of Medical Affairs with Therapeutics, a biotech company based in the United States, and I live in Minneapolis, Minnesota. And I'm here to talk to you today about our Phase 2 study for Ublituximab in relapsing MS. Ublituximab is a novel glycoengineered anti-CD20 monoclonal antibody. Now, what makes Ublituximab different from other monoclonal anti-CD20 antibodies that are currently either in development or approved for the treatment of multiple sclerosis are two things. One, it recognizes a different epitope on the surface of CD20, which defines the identity of the B cell. And secondly, and probably most importantly from a clinical perspective, is that Ublituximab is glycoengineered, which means that certain sugar molecules are removed on the tail end of the antibody. And this allows for an enhanced interaction with the natural killer cell through the CD16 receptor. And where we think this may offer some advantages clinically is enhanced potency of B cell depletion through a process called antibody-dependent cellular cytotoxicity. That enhanced potency could result in shorter infusion times and lower doses. And in fact, there's some published data showing that the ADCC activity of Ublituximab is approximately 100-fold greater than that of rituximab, for example. So we think we may have potency, which again would give us some clinical advantages. Okay, here you can see the study design. This is a one-year placebo-controlled study. The placebo phase only lasts for the first month of the trial. So patients who are randomized initially to placebo receive 2 infusions of placebo at day 1 and day 15. And subsequently, they're monitored for 2 weeks after that. After that cumulative 1-month period, they're unblinded. They're told that they were on placebo initially, and then they start the trial all over again, but on active treatment. If you look to how the top of the poster, where you can see the organization of the cohorts, there are 6 treatment cohorts which vary in terms of infusion time and dose. So what we wanted to figure out in this study was the optimal, best tolerated infusion time, and we wanted to make sure that our antibody was working in MS patients. So prior to this point, there had been approximately 500 oncology patients, patients with various B-cell malignancies who had been treated with the drug. So we had a pretty good idea of the safety and tolerability of Ublituximab going into this trial. We just wanted to make sure that it one, worked in MS patients, and 2, that we could come out of this trial knowing what dose and infusion time would work best. So with that as a background, let's go into the results here. So you can see the patient population. There were 48 patients randomized, and you can see the mean age and disease duration, skewing a little bit on the older side, but otherwise pretty typical of a relapsing MS population. Now in the panel next to it, you can see our primary endpoint, which is B-cell depletion. So what we were aiming for was a 95% B-cell depletion at 1 month, which was the time of the first 2 infusions plus 2 weeks. And what we got as a point of fact at that point was greater than 99% B-cell depletion. So we hit our mark and then some. In fact, we reached our 95% B-cell depletion mark after the first infusion. So that told us that essentially we had potency to spare with Ublituximab. Now that was our primary endpoint. We knew that we hit our primary endpoint. Now the other measures, we're looking at the typical MRI and clinical measures that you would have in a phase 2 trial. So with that, looking at our MRI data, gadolinium-enhancing lesions, there was a mean of about 3.6 GAD-enhancing lesions at baseline. By 24 weeks, there was not a single detectable GAD lesion in any of the patients, and that persisted at week 48. So to this day, we have seen no GAD-enhancing lesions in any patient. So effectively a 100% decrease in GAD-enhancing lesions. Similarly, with T2 lesion volume, we see a decrease at 24 weeks, which continues to a further decrease at 48 weeks, a cumulative decrease of about 10% in T2 lesion volume. Looking at the other clinical measures, we had an annualized relapse rate of 0.07. So 97 or 93% of the subjects were relapse-free at the end of the study, when in the year prior, there was an average of about 1.5 relapses per subject. Cumulatively, looking at EDSS and all of our clinical measures, we have 74% of the patients that have achieved NITA status at 1 year. Looking at the safety, the most common adverse events were infusion-related reactions. 24 out of 48 patients, or 23 out of 48, so 48% of the patients had one or more infusion-related reactions, but bearing in mind that we were testing out as fast of an infusion as possible, so we're bound to get a few more infusion-related reactions. That said, we did not have any patients that discontinued the study due to any adverse events with Ublituximab. So tolerability and safety look very good at this point. So by way of conclusion, we hit our endpoints. We hit our targets in terms of B-cell depletion. Safety and tolerability looked very good, and the initial clinical measures look very good at this point. So with this, in terms of next steps, what are we doing next? Well, we're taking this as a reference point, and we're moving forward with our phase 3 trials. So we have a dose in mind and an infusion time in mind, and we currently are fully enrolled with our phase 3 program. We have 2 phase 3 studies ongoing now, the Ultimate 1 and 2 studies, and we're expecting a readout by the end of 2020. So although I work in industry now, I started off my professional career in basic science, as a basic scientist. I'm trained as a basic neuroscientist, and what motivated me to go into that field was to improve the lives of people living with neurological diseases. And fast forward to now, I think those of us in industry, those of us who are clinicians, and the patients and patient advocacy groups should be working together, all for the same goal of improving the lives of people with MS. And with that, I want to express my sincerest gratitude to the participants in the trial. We really couldn't have done it without them, and we couldn't move this field forward without your help. So thank you.